Hello, I'm Jim Cornell, and this is Base to Base, the weekly biotech podcast from Air Coastal Media. Yes, welcome again to the Base to Base Biotech podcast, and this is number 19, going out on July the 25th. It is Drowning Prevention Day, and it is also Red Shoes Day. I'm actually recording this with no shoes on at all. Before we get going, today's podcast is being sponsored by NIPRO. NIPRO Medical Europe is part of NIPRO Corporation Japan, a leading global healthcare company established in 1954. With over 40,000 employees worldwide, NIPRO serves the medical device, pharmaceutical and pharmaceutical packaging industries. NIPRO Medical Europe is a global market leader offering a comprehensive portfolio of medical disposables and machines across 6 divisions: renal, hospital products, diabetes, cardiopulmonary, enzymes, and in vitro diagnostics. With a worldwide footprint of manufacturing sites, sales offices, and distribution centres, NIPRO Medical Europe services the EMEA region. From hospitals to home care, NIPRO is committed to providing the highest quality products that improve the lives of patients and meet the needs of healthcare professionals and procurement managers alike. NIPRO recently launched LiniXia, its new reverse osmosis system that reliably ensures safe and pure water for every dialysis session. With LiniXia, NIPRO has added an entire range of water treatment solutions to its renal care portfolio, combining innovative RO technology with user-friendly controls, detailed logs, and remote supervision capabilities, all while ensuring up to 90% yield of high-quality, ultra-pure water. If you want to know more, visit www.nipro-group.com/renal-care. Alright then, on with the podcast, and it was another very busy week of news. Some of it we will get to shortly, and if you do have news or suggestions for who should be on the podcast, or indeed any other suggestions or comments, please do get in touch at jim@deeptechdigest.com, which is also the address you can use if you're interested in sponsoring a podcast or advertising with us. And I'm glad to say that lots of people are contacting us, so thanks so much for that. Last week we talked about neutrophil extracellular traps, otherwise known as NETs, and their potential as a new therapeutic class being developed by Citral, with Citral CEO Eduardo Bravo. And we also had a conversation about, among other things, antimicrobial resistance and using AI to develop new drugs, with Ampli Discovery COO Dermot Tierney. This week we're talking about NIPRO's recently launched LiniXia, which is its new reverse osmosis system to ensure safe and pure water for all dialysis sessions, and that is with Jesus Garcia, NIPRO Product Manager, Water Treatment. And we'll also have a conversation about the future of biologics manufacturing with cell-free protein synthesis with LenioBio CEO André Goerke. Right, first to this week's news that you may have missed over at deeptechdigest.com. The FDA granted orphan drug designation to a new gene therapy for ALS. Elix and Link combined on a new drug discovery platform, and Sanofi is acquiring vaccine biotech Vice Bio for $1.6 billion. Bakar Labs is set to build a new R&D facility for biotechs. Johnson & Johnson received a priority review for its high-risk non-muscle invasive bladder cancer treatment. And we had an article on how cells repair toxic DNA damage linked to cancer and premature aging. New molecular degraders are offering hope in the fight against aggressive breast cancer. Foremost demonstrated a new approach to molecular glue discovery, and LG debuted next-generation precision medical AI. Johnson & Johnson is aiming to revolutionize plaque psoriasis treatment, and you can read all of these and plenty more at deeptechdigest.com. Alright, time for the first of today's guests. NIPRO recently launched LiniXia, its new reverse osmosis system that ensures safe and pure water for every dialysis session. We had an opportunity to have a conversation with Jesus Garcia, NIPRO Product Manager Water Treatment, about the new product and a whole lot more. Thanks for joining us on the podcast today. First, I wonder if you could tell us a bit about NIPRO.
Yeah, sure. NIPRO is a global healthcare manufacturer and solutions provider, which is based in Osaka, in Japan. We were founded in the 50s, and what began as a local endeavor has grown into a truly global company. So today we are nearly 40,000 people delivering life-sustaining solutions with a structured presence around the world. Our portfolio spans renal care, hospital products, vascular and interventional therapies, and pharmaceutical primary packaging as well, all united by one guiding principle, which is Sanpoyoshi. Sanpoyoshi is Japanese philosophy and means three-way satisfaction. So it's a way for the patient, for the society, and also for us. That is not just a slogan, it is how we measure success. In short, we combine the Japanese precision with a global reach to help build a better standard for care. One innovation, one patient, and one partnership at a time.
All right. What are some of the current issues with water in dialysis?
Well, water is, as very well said, is an issue. Water is the foundation of dialysis therapy. So it presents two major challenges today: availability and quality. Only a very small fraction of the Earth's water is accessible and suitable for medical use. In dialysis, where ultra-pure water is essential, disparities in water quality across regions can compromise treatment safety and consistency. This makes a robust, reliable water treatment system not just a technical requirement, but a clinical imperative.
And could you tell me about the latest addition to your dialysis portfolio?
Of course, our newest innovation is called LiniXia. It's a reverse osmosis water treatment system designed specifically for dialysis environment. Certified under the latest European medical device regulations, LiniXia is engineered to deliver high-performance water purification while optimizing operational efficiency. It reflects our commitment to sustainability, digital integration, and user-centric design, and arrives at a time when dialysis clinics are under increasing pressure to do more with fewer resources as well.
How does this improve on previous methods?
So the device, LiniXia, represents a leap forward in usability, automation, and sustainability. It features an intuitive interface that simplifies a lot daily operation in maintenance, mainly reducing the learning curve for clinical staff and healthcare professionals. Technologically, it integrates advanced monitoring and control systems that enhance reliability and traceability. From a sustainability perspective, it supports resource conservation, a growing priority in healthcare procurement and tender evaluations.
And how was it developed?
So LiniXia was co-created with the field. We conducted extensive interviews with dialysis professionals across Europe and beyond to understand their pain points and expectations. So their feedback directly shaped the system's design, from interface layout to maintenance workflows. And internally, our R&D department and technical service teams collaborated as well to ensure that the solution was not only compliant and robust, but also adaptable to diverse clinical settings.
All right. Now, it was launched at the ERA 2025. What was the reaction like to the launch?
So the reaction was very positive from my point of view. The launch was met with enthusiasm. Healthcare professionals across Europe expressed a strong interest and several place orders during the event, by the way. Even competitors were having a look around and took note. So I think that's a clear sign that the LiniXia is setting a new benchmark in dialysis water treatment. The positive reception confirms that we are addressing real needs with a solution that's both timely and forward-looking.
Is it available globally?
Yeah, we are just launching it in Europe as a first step, but available all around the world. We are making the promotion everywhere. Yeah.
Oh, good. What else is in your portfolio?
Well, in NIPRO offers a comprehensive dialysis ecosystem, full portfolio. So beyond water treatment, it includes dialysis machines, a wide range of dialyzers, including high flux and super high flux membranes, vascular access solutions, and digital tools like NephroFlow for treatment management. We also provide patented loop systems, media panels, and full project support from design to technical service, which is really, really important in water treatment areas. This is end-to-end capability allows us to deliver integrated, high-quality care solutions tailored to each facility needs.
How often do you either develop new products or evolve existing products?
So at NIPRO, innovation is not a buzzword, it's a duty. A duty grounded in our Japanese heritage of precision and brought to life by our global teams who touch countless lives every single day. With more than 40,000 colleagues across the world, we are fully aware of the responsibility we carry. So we deliver life-sustaining therapies to hundreds of thousands of people. Their well-being, even their lives, depend on the quality and integrity of our work. That is a responsibility we do not and will not take lightly at all. Whether it is LiniXia, LiniXia or NephroFlow, each product we launch carries the DNA of Japanese craftsmanship and is refined through real-world insight gathered from our IMEP medical education platforms. Across our global training centers, we actively engage healthcare professionals, collecting feedback that goes straight to our R&D pipeline. This proximity fuels relevance. It is how we ensure that we are solving real problems and not imaginary ones. And our focus doesn't stop at renal care and dialysis with innovations like Makoto, which are our AI-enabled imaging solutions, and Aperta PTA, our next-gen angioplasty balloon. We are expanding into vascular and interventional fields as well, always with the same mindset to create a new global standard in care. But innovation is without integrity is just a risk. So this is why governance and compliance are not peripheral to NIPRO. They are central. Compliance is not a checklist. It is not negotiable, the heartbeat of our company. We are strengthening our internal frameworks, investing in training, and embedding a culture where doing the right thing, even when no one is watching, is simply the only way we operate. This is how we honor the trust placed in us by patients, professionals, and partners alike.
In terms of if you put one of the LiniXia into use, how many people would that serve?
So it depends. There are many different sizes of clinics. You can have one clinic. So every clinic has one water treatment, and it really depends. We have small clinics up to starting from 5 patients, for example, and up to 100 at the same time. So we really need to ensure that water quality and the device is running perfectly with a minimum downtime, of course.
I would imagine that different countries have different needs as well. I mean, it's all right saying, in the US or the UK, but then you've got countries where water is more of an issue.
Yeah, it's really an issue, as I told you, because of accessibility. In some countries, they don't have water to make dialysis, so it's really an issue, and because of the quality of the water. And regarding the different countries and different needs, fully agree. Indeed, they are sometimes regulated in a different way, so the devices need to meet different local regulations sometimes. So yeah, although it's expanded all over the world, sometimes it's not easy to match all the regulations at the same time. and some particular needs need to be addressed in every market.
Is that how you work? Is you kind of work with the clients to figure out what they need and then you address that?
Yeah. So it's very difficult because it takes time to develop and to take the approval to be sold and so on. But this is how NIPRO is trying to evolve and move forward. So we have products today, but we always, once we sell, we are keeping the contact always with the customers. Is it okay? Do you like it? What most do you like it? What could be improved? So in our future, we can take it into consideration for improvements.
Well, that's excellent news for anyone needing dialysis treatment, and it's also great to hear about the other technologies NIPRO has and is developing. From a Japanese company, we're now going to Germany to talk with LenioBio about its ALICE platform and the future of biologics manufacturing with cell-free protein synthesis. Our conversation is with LenioBio CEO André Goerke. All right, to get things started, first of all, thanks for being here. And I wonder if you could tell us a bit about the company.
Yeah, sure. So, as you know, called LenioBio. We're a biotech startup from Germany, located in Düsseldorf, with our administrative offices and have a pilot plant in Aachen. The company has been founded in 2016, has been founded by Dr. Roberto Martis, who had the vision of creating, that is still our vision, right, to create better access to medicines for those people that are in need of it. What we do is we basically enable life science innovators to produce those proteins that are super difficult or impossible in conventional protein expression systems. So that the innovators, we can support the innovators to develop the medicines of the future, if you want. For that, in order to help them with that, what we have done is we have invented a pretty unique protein expression system that makes it extremely easy to develop and produce extremely difficult proteins. That's basically us in a nutshell.
So could you maybe go into a bit more detail on what ALICE is and how it works?
Yeah, so the ALICE is actually, the name is an abbreviation. Right? It stands for Almost Living Cell-Free Expression, and that joined up nicely to ALICE. It's a system, it's a lysate actually. It's a yellow liquid, it's a lysate, and it's a system to express the most complex proteins without a cell. And we have developed this for over 8 years now, and I think we can say that we have reinvented cell-free protein expression by now. Cell-free has been here for decades, but cell-free always had a lot of limitations, and that is what we worked on to remove. So it could always produce proteins, but only the simple ones. Our system, ALICE, it does the most complex proteins, and it also does non-natural ones designed by AI, for example. Then the other thing with cell-free systems was or is they produce generally, when they produce complex proteins, they produce them in minor quantities. We have taken the system and the system now produces grams per liter, right? So comparable to conventional systems. And the other thing is the system, cell-free systems, most of them, but one apart from us, they were not scalable. And we have taken the system and now scaled it approximately by a factor of 200,000. We stopped here, but we have a possibility to go beyond that, and I can talk a bit about that later. So the result, to come back to your question, the result is a cell-free system that combines basically the best of both worlds. Right? It just has the freedom of a cell-free and the capabilities of a cell-based systems. And what we use to do that is a high-performance BY2 cell line. BY2 is a plant cell, right, that comes from the root of a tobacco plant. And we take that cell line and we transform it into a cell-free lysate. And at the end, what you do have is you are left with the protein expression machinery without the limitation or the constraints of a cell.
So what are the applications for eukaryotic cell-free proteins?
Eukaryotic is something, I realized, going into this, that it's not always straightforward for people to understand, okay, what does that mean? Eukaryotic, prokaryotic, what are the differences here? And maybe a quick explanation to that, right? So eukaryotic, basically what it does, it refers to organisms that has cells that contain a nucleus and some membrane-bound organelles like plants and mammals have. Compared to bacterial organisms, right, who are prokaryotic, those eukaryotic systems, they're able to generate proteins that have, for example, post-translational modifications. These post-translation PTMs, the post-translation modifications, they are often very, very relevant for the function of the protein. So that means a eukaryotic system such as ours can produce the most complex proteins. There are very few limitations of what we cannot do. The established protein expression systems, the conventional ones like CHO, right? For example, they are eukaryotic as well, but they're cell-based. So they have to basically balance the protein production with cell growth and division, and that limits their ability for protein synthesis, especially for toxic and non-natural proteins. Cell-free systems like ours, they directly apply all resources purely towards protein production. So when you remove that constraint of a cell, the freedom to design new proteins is almost unlimited. Especially now where protein design is being transformed, through generative AI, the ability to test and produce novel designer proteins is super important, right? What you have to do after you tested all of them and designed them in silico, you need to generate real life data and feed it back into the generative algorithms to make them smarter. And that is one application, for example, that we have to come to your question, right? So our system is very fast. It takes 24 hours, right, to express a protein. So that means when you actually have your designer proteins and you want to test them, you need a system that doesn't constrain you and lets you do it. And second, you need to get the data fast to feed it back, because what you really want is to make sure that your algorithms do not hallucinate. So that is one application, but there are more than that. Our focus is generally very difficult to express proteins like multispecific antibodies, like novel vaccines, like previously inexpressible drug targets. I think these are the main applications, but there is no real limit. There's also not a limit to applying this only into the pharmaceutical space. Basically, you could apply it into the animal health and into the crop protection space, but our focus is pharmaceutical, right, and human medicine.
You've obviously done a lot of work in this area and made a lot of advances. What were the challenges initially in this?
Yeah, there's still a lot of challenges left, right? But we tackled a few along the way. The challenges were obviously to remove the limitations that a cell-free system has, right? So our view was to make this extremely simple and extremely powerful, right? So we tried to come up with an all-in-one system. So one that we would not be reconstituted, where you take the several ingredients and put them together to a specific experiment, then you run that one experiment. We made a one-in-all lysate. The only thing that you do is you take the lysate, you add the DNA, and then you wait. So that was one of the challenges, is to actually bring the different components together and say, hey, we make a one-in-all solution. The other one is in the process. How do you take the cell and you remove everything that works against or limits protein production and leave everything that is essential for the protein production and for the folding and the microsomal activity of the lysate? So there was a very, very fine line to balance between keeping the one and removing the other. And then obviously, once you have something running in the lab, it doesn't mean that you actually can apply it. So we had to bring this from the lab bench, pipetting something, into industrial equipment, which we did. So it took the team over, I think it's almost 2 years to actually make that tech transfer from the lab into that. And these are just the internal challenges. And external is exactly what I just mentioned to you, that creates an idea in people's minds, in our customers' minds, of where are the limitations of cell-free. And that was probably at the beginning as well a bit of a struggle to say, no, we do understand that is your experience with cell-free, but that is precisely what we have worked on. So you can take our system and you can scale it all the way from your first experiments up to your tox studies. We are not GMP yet, right? But that is what we're working on. So we actually are in the space of discovery and development. So I would say these were the key challenges. By now, we actually can demonstrate, we have demonstrated that we can produce the most complex proteins, and we also can scale them. We had a number of things, that are also done by not us, but customers where they produced drug targets that they had never expressed before, right? And we are obviously very happy that we can help with these breakthroughs.
Because a lot of companies are working in an area that's very already set, and so you don't have to explain anything. But clearly, when you are doing something new like this, then you've got that extra challenge of communication.
Yeah, we have started to work with partners that are very innovation focused, like CEPI, for example. And we also will start doing the same thing with HERA, right? And before we have worked with Horizon in the EU. We have a stage to display, what the technology does. This is one aspect. The other aspect is we have entered and we are still there on the life cycle phase very early on. Because once you're set on your protein and you want to produce this as a biopharmaceutical medicine, you will not switch platforms anymore, right? So we start very early in the game where you really start with screening, because also here, your success rate with a system like ours is significantly higher than if you do it with a conventional cell-free system, just by the bandwidth of proteins that you can do. So we start in the screening, we support lead optimization thereafter, and then scaling these quantities for assays so that you start generating experience with the system, with its simplicity and what you can do. That is the second aspect. And the third, which is what we're doing now, we're looking for collaborations. We have a few, but we're looking for more collaborations with more established partners that actually complement our own ecosystem, right? So that we are a reliable partner to the innovator community.
How does that relationship work? What is it that you provide to companies?
Some of these instances are co-development activities, right? And others are more going to the direction of co-commercialization. What we provide to them mostly is the ability to access the inaccessible at the moment, right? So the access to proteins that is not possible with traditional systems. And I think that is also part of the way that we see ourselves. We do not see ourselves replacing things like CHO. We see ourselves as the mechanism that you use to open market segments that you currently cannot access. I mean, you take a lot of the starting with drug targets, right? If you actually want to test your therapeutic, you need to get access to the target first, and you have a lot of targets that are membrane or transmembrane proteins, right? Super difficult to express, if at all, because lots of them are toxic to the cells, and therefore lots of them are structurally not characterized. What we can offer to the companies here is we do these with ease for you at relevant quantities so that you can test the therapeutic. The other area is, of course, the area of multi-specifics, right? If you think about different elements you need to engineer together in a multi-specific antibody, right? It takes a lot of cell line development, and it takes a lot of bioengineering thereafter in order to bring those together and make them a viable production system. For us, as it is an open system, you do not have constraints. So you take different pieces of DNA, add them together, and express it also in a very straightforward fashion. So meaning apart from the access, we also offer you simplicity. And then you have the part of vaccines. Vaccines and we all saw that during the pandemic, has a significant challenge when it comes to preparedness. And here, an agnostic system, our system is protein agnostic. That means the system doesn't care if you today produce protein A, tomorrow produce protein B, and thereafter you produce protein C. That is different with a cell line. A cell line you do develop for a certain protein. So you have a long lead time. And when you're done, you will produce that one protein. So difficult for pandemic preparedness purposes. In our case, what we offer you is an agnostic system. Number one, our system is great for protein-based vaccines, needless to say, right? We have a proprietary mechanism where we have a platform, VLP platform with a click chemistry, that you can site-specifically decorate with antigens. So that gives you a lot of freedom. Right? Basically, you can produce the lysate wherever you want, because whenever you want, because it is not protein specific. So you produce your lysate over the year, you store it away, you take it out when you need it. Second thing is you could even do that with a carrier, which is the VLP, right? So you can pre-produce the carrier once you know, okay, this is the vaccine that I'm after. And then, the last thing, which is the antigen, whenever you need to decorate it with a different antigen, that is the only thing that you then add differently to your system. So pandemic preparedness within 100 days, because obviously there are steps after protein expression purification, like the fill finish and the distribution. But that vision of, yes, you can actually tackle a pandemic in 100 days before it becomes an epidemic event can become reality with this.
Right. I was going to ask you about the pandemic and how that shaped both the field and your company. Has it made a massive difference?
We are actually working with CEPI on this. CEPI has launched its mission for pandemic preparedness of below 100 days. We have received a grant from CEPI. We have been working, over the last, 16, 17 months now with CEPI, made great progress here. We'll continue to invest into this together. So yes, I expect us to make a real difference here going forward. And it is something where we are starting to see increasing interest in our technology.
And you also have other EU projects. Could you tell me about some of those?
Yes, actually, we have not announced this yet, but we will do so this week. So, which is the HERA grant that we just obtained. So we have, which is also an EU sponsored program. The HERA grant, what it will do is it will give us the possibility to develop the further scaling and optimization of our system, and meaning of our production system to produce our lysate, right? So that we really can get this into the next step of scaling, into the next step of also making the process super efficient, right? So that you can easily tech transfer this into GMP environments thereafter. And that is what we're going to now launch and work on. We have received the STEP seal, which is a seal that you receive when you're considered a strategic technology for a specific field within the EU and Europe. Right? And we're very happy about that, so that we within Europe also can really make a dent and contribute to things such as pandemic preparedness and others.
Does that involve working with other organizations and companies, or is that just you continuing to do what you do?
In most of these projects, we do have partners that work with us. I think, in the HERA, we will now, obviously, we will develop this, but in the CEPI, we are working with companies, for example, like Touchlight that do synthetic DNA. We work with universities and institutes like Wageningen, which is close by, for animal testing. So it is always good to connect our technology to ecosystem technologies, right? So that you can amplify it.
How do you see the evolution of biologics manufacturing?
I think from what I see, and that is not only my opinion, it is, you see that in conferences at the moment, and you also hear from customers. Cell-free systems are clearly rising, and I'm convinced they will become an established platform next to the existing mammalian cell-based systems. Because what they are doing, they will open market segments that you cannot access currently. And they will be a key tool, for the digital protein design and testing revolution that is occurring. As I mentioned before, they have the power to feed algorithms with data fast, so that the functional new proteins will be designed that can tackle diseases in a much more tailored way. And that you can do this at the moment and still efficiently. But I'm obviously a bit biased, right? But I'm convinced that cell-free has come to stay and has a lot to contribute.
Are there still any challenges remaining?
Yeah, I mean, look, like with every new platform, regulations part, right, and the related standards, the classification of cell-free and how to, handle it in that context, they have to be shaped further, which will help us as a company, that you have a framework that is as established as with the existing technologies. Scaling and efficiency have further room to improve, which is what we are working on. But I would say the starting point to do so is already better than it was with traditional cell-based systems back in the past, right? So we are already to start with at grams per liter, which is a pretty good starting points. And the technology to improve that is there already. It doesn't need to be invented. And last, I would also say we need to nurture the innovation appetite for new platforms in the innovator and in the investor community, because there's always a lot of hesitance, when you go into something new. We need to do our best to de-risk this, and make sure that the people test these new tools that gives them really new opportunities to serve their markets better.
And so what does that mean for your company? Where do you go from here?
Where we go from here is we go into strategic partnerships with innovator companies. That's our clear aim. With the scaling of the technology and with efficiency improvements, we definitely want to create access through licenses, to pharma companies so that they actually can utilize our platform to produce innovative medicine, right, on it.
And I assume as well that because knowledge is increasing, that you probably don't have to maybe do that communication piece as much as you used to as it grows.
By now, we have started to have the right conversations, right? And we are enlarging the community that is working with us. So that is super helpful because that helps us obviously, on both sides to generate a lot of data that helps us to prove the concept and show this, right? So this is increasing. We hope to do a lot of publications also over the next months and years with this that are self-speaking. We're quite happy that we have a few, and that therefore the appetite to utilize it at actually every stage of drug discovery and development increases. We are in a good place. It's going to be obviously, like for every startup, still a lot of work, especially also to finance our activities so we can run fast. I'm very positive on the future of LenioBio here.
It's really interesting learning about something that really has the potential to make a big difference. Kind of took me back to my early days studying plant science and learning about eukaryotic cells. Quite a lot has changed, I admit, since my first degree back in the last century. Almost feels like the 19th century at times. Other things have also changed since then, including my hairline and my waistline. Anyway, that's it for this week's interviews. If you have questions or comments, suggestions for guests, or want to sponsor one or more podcasts, please do get in touch, and you can do so at jim@deeptechdigest.com. Finally, a reminder that this week's podcast was sponsored by NIPRO. NIPRO Medical Europe is part of NIPRO Corporation Japan, a leading global healthcare company established in 1954. With over 40,000 employees worldwide, NIPRO serves the medical device, pharmaceutical and pharmaceutical packaging industries. NIPRO Medical Europe is a global market leader offering a comprehensive portfolio of medical disposables and machines across 6 divisions: Renal, Hospital Products, Diabetes, Cardiopulmonary, Enzymes and In Vitro Diagnostics. With a worldwide footprint of manufacturing sites, sales offices and distribution centres, NIPRO Medical Europe serves the EMEA region. From hospitals to home care, NIPRO is committed to providing the highest quality products that improve the lives of patients and meet the needs of healthcare professionals and procurement managers alike. NIPRO recently launched LiniXia, its new reverse osmosis system that reliably ensures safe and pure water for every dialysis session. With LiniXia, NIPRO has added an entire range of water treatment solutions to its renal care portfolio, combining innovative RO technology with user-friendly controls, detailed logs, and remote supervision capabilities, all while ensuring up to 90% yield of high-quality, ultra-pure water. If you want to know more, visit www.nipro-group.com/renal-care. So, until next week, wherever in the world you are, I hope you have a great week. Thanks for listening, and I hope you'll join us again next week for another Base to Base Biotech podcast. The Base to Base Biotech podcast is an Air Coastal Media Limited production.